Welcome to the final video in our microbial genetics series. This video is going to be all about horizontal gene transfer. And in a nutshell, horizontal gene transfer means that a bacteria is acquiring DNA not from the parent cell. So remember, bacteria divide by binary fission. So essentially, imagine a cell just kind of elongating, being stretched out until it pinches into two separate cells. And before the actual splitting happens, the original cell must copy its DNA. That way, when the cell splits into two, each of those two cells has a copy of DNA. That's considered vertical gene transfer, kind of like when, right, you have similar DNA to your parents. That's vertical transfer. And if you have a child and you pass your DNA, again, vertical transfer. So horizontal means it's not coming from like a family member, if you're thinking of it at the human level. So in bacteria, because they do asexual reproduction, if a bacteria acquires DNA not from that original parent cell that made it, we call it horizontal gene transfer. So there are three types of horizontal gene transfer that we are going to talk about: conjugation, transformation, and transduction. So these are the pictures we're going to use. Nice little summary slide. So what I recommend before the exam or any of the assessments, you should be able to look at this picture and kind of describe what is transformation, what is transduction, okay? So let's start with conjugation. So I used a different picture here, but the same gist. And essentially what happens is these two cells that you see right here actually start off as separate cells, okay? But notice one has what's called the F plasmid. That's what it means by being an F plus. The F plasmid stands for fertility plasmid. And basically, that plasmid that you see here, remember, a plasmid is a circular piece of double-stranded DNA that is separate from the chromosome, okay? The information on this plasmid is not essential, not required for the bacteria to survive. It's just kind of offers a bonus feature, okay? And in this case, the fertility plasmid codes for a pilus. We learned about that when we talked about cells. And the pilus acts kind of like a fishing line. So originally, like I said, these two cells are separate from one another, and they can actually be quite a distance away. And so this F plus cell, the one with the plasmid, is able to elongate or extend out the pilus so that it can go out and grab hold of a neighboring cell that does not have this plasmid. So imagine if you're sitting on the shore of a pond or something, and right, you cast out your fishing line, that fishing line goes way out, and ideally it's going to hook onto a fish, right? So in this case, the pilus is casted out and hooks onto another cell. Well, what do you do if you get a fish? You reel it in, right? You bring that line closer to you. And so that's what the pilus does. Once it's grabbed hold of another cell, it's going to bring it in so that they're close together. And that's what you see here in this top picture. And so then what happens is they form what's called a mating bridge. And so notice now it's hollow in between. So this mating bridge is kind of like a little tunnel connecting the two cells. And notice what's happening in this picture. Do you see how this outer strand, remember, because a plasmid is double-stranded, so each of those circles is one strand of nucleotides. That outer strand is cut, and it's feeding that strand through to this F minus cell, or F negative cell, however you prefer. And so the reason this happens is once it's done, it transfers that entire outer strand of nucleotides to the F negative cell. And what do you think is going to happen? DNA replication. The cell is able to use the one strand that each of them has and create an anti-parallel complementary second strand. So at the end of the day, both of these cells now have a double-stranded plasmid. So now notice they're both F positive. Now they both have this fertility plasmid. Now, as you see in this picture, this other cell can now form a pilus as well, and then go and share this plasmid with other bacteria. So again, this F negative cell has acquired DNA from a neighboring cell. So it's horizontal gene transfer. All right, the next one is transformation. And this is where there's no cell-to-cell contact. Okay, another cell isn't even really involved. So what we have is this cell here, and it's just out here minding its own business. And notice there's a piece of DNA just hanging out in the external environment. Now you might be thinking, Chelsea, why is there DNA just chilling in the environment? Well, maybe a neighboring cell died, okay? And when a cell kind of, it can explode, it can lyse. And in doing so, right, all the pieces of the cell are going to go into little bits and kind of be spread throughout the environment. So sometimes that means there are pieces of DNA just hanging out. And some cells, not all, only cells that are called competent cells are able to just take that DNA in from the environment. Okay, so this is not an easy thing to do because DNA is a pretty big molecule, I guess, and it's charged. DNA has a negative charge, so it's not really compatible with the plasma membrane because remember, the plasma membrane has a hydrophobic barrier, and so this isn't easy. Only special cells, again, competent cells, have a mechanism in place where they can bring this DNA in. Now, normally, the only way this can happen is if this DNA came from a cell of the same species. So if this cell here was E. coli, then this bacteria likely came from a dead E. coli cell because they're kind of compatible, or at least related evolutionarily. I think I just made up that word. All right, but the point is this cell is here minding its own business. It's, detects some DNA outside of the environment. Because it's a competent cell, it's able to bring that DNA in. And because the DNA came from a related or similar organism, it's able to actually incorporate that plasmid or that extra piece of DNA into its own chromosome. So that's what you see happening down here. Okay, so in a nutshell, a competent cell takes DNA in from the environment and incorporates it into its own chromosome. Pretty snazzy, huh? We use this actually in the lab. When I was doing research in graduate school, you're able to force cells to become competent. If we expose them to a little blast of heat, the heat kind of makes them go, whoa, and it opens them up. It makes them more porous, and they can bring in DNA. And that's how we're able to figure out what certain proteins and things do, is by kind of forcing a bacteria, something that's safer for us to work with, to take in that DNA. It'll then make that protein, and we can study it. So I did this a lot in the lab. But why transformation is super important is because there was a guy named Griffith. Please write that down, Griffith. And what Griffith did was he's the one who first showed that horizontal gene transfer exists. And how he did that was he had this bacterial strain, and it was non-virulent, meaning it was safe. It doesn't make animal or people sick. And when they injected this mouse with this strain of bacteria, the mouse was fine. Just like I said, it was not a pathogen. All right, so that's kind of what they expected to see. Then they took the strain and they exposed it to heat, which killed it. Okay, so the heat-killed strain is going to be injected into the mouse. Well, because it's dead, right? The mouse was fine. Okay, and this was a pathogenic version. I should have mentioned that. So, because notice, see how this one has a capsule around it? So this is a pathogenic strain, and they exposed it to heat to kill it. So of course, the mouse was fine because the pathogen was dead. Now, what they did is they mixed the safe non-pathogenic bacteria, which is in blue, and they mixed it with dead pathogen that they exposed to heat. Okay, so what they expected to happen was the mouse would be fine, right? Because the pathogen was dead and the living bacteria was safe. But what surprised everybody was the mouse got sick and died. Okay, and so this was the first evidence that maybe these, living safe bacteria picked up some of the bacteria from the dead ones. And by picking up some of that DNA, it allowed them to become pathogenic. And that's actually what happened. When the pathogens were killed by heat, it allowed some of the DNA to get out into the environment. And the safe non-pathogenic bacteria took it up and they acquired the capsule, that sugary layer that a lot of pathogens have. And so when they acquired that capsule, they then became pathogenic themselves, and that's why the mouse died. And then what they did is they were able to go in and they isolated the virulent strain and inoculated it into the mouse, and it died. So it was just showing that the bacteria had in fact become pathogenic by acquiring DNA from the dead pathogenic bacteria. Pretty snazzy, y'all. All right, last but not least, why did that not change? Hold on a second, sorry, there we go. All right, the last one is transduction. So with transduction, this one we're just going to kind of touch on a little bit right now because we're going to talk about viruses in our next series. So we'll kind of circle back to this. But there are certain types of viruses that can only infect bacteria, and we call them bacteriophage, which is this word you see here. And so viruses contain genetic material and a little protein coat that protects the genetic material. And when a bacteriophage attaches to a bacteria to try to infect it, it injects its DNA like you see here. Okay, now normally this is viral DNA, and then the bacteria would get sick, much like we do, right, when a virus infects us. But what can sometimes happen is when the virus is going through its replication cycle and making lots of other viruses, sometimes the virus gets confused, and instead of picking up viral DNA before it leaves the host, it accidentally picks up pieces of bacterial DNA. So then this virus is holding on to bacterial DNA. So when it goes and infects another bacteria, instead of injecting viral DNA, it injects bacterial DNA into the bacteria. And so the bacteria doesn't get sick or anything, but it does acquire whatever DNA was on that piece, whatever was in that virus. So when we get to the diseases in Unit 4, we are going to talk about some pathogens that get their toxins from this process. And if they actually don't if they haven't gone through this process, they're actually not dangerous. It's when the virus accidentally introduces a bacterial toxin into the bacteria that's going to make it pathogenic. Okay. Now, again, once we go through the virus life cycle, this is going to make a little bit more sense. So again, I will circle back when we get to that conversation. But for now, focus on keywords for each of these horizontal gene transfer. Okay, so conjugation involved the pilus, which brought two cells together. They formed the mating bridge. They shared that plasmid. So at the end of the day, they both were F+ cells. With transformation, we talked about just a cell that came into contact with DNA. It was just in the environment. If it's a competent cell, it can take in that DNA and then incorporate it into its own genetic material. And then with transduction, this involves bacteriophage that infects the bacteria, but instead of injecting viral DNA, it injects bacterial DNA that the bacteria can then use. And sometimes this offers an advantage, like a toxin or something. All right, y'all, that is the end of our microbial genetics conversation. Please, please, please let me know if you have any questions.